This lesson is going to be the first of probably 3 lessons about the cell membrane. Now remember, the job of the cell membrane is to regulate what is entering and leaving the cell. We're going to talk about some properties that the cell membrane possesses that make it possible for it to do that. The one thing I do want you to understand as we talk about the parts of the cell membrane is that it is not a solid structure. It's not like a piece of plastic or something like that. It's not even like a piece of mesh or screen. It's actually fluid. The parts that make up the cell membrane are not physically attached to one another. They're floating around, which is kind of bizarre to think about, but they're actually able to move around. So remember, the cell membrane is made of these things called phospholipids, which are 2 long fatty acid chains with an attached phosphate group. And you know that what a phosphate group looks like, and you know that phosphates are charged, they're polar. So the head, we call it the head, the phosphate group, is hydrophilic because it's charged and it's polar, it can dissolve in water. The 2 fatty acid tails, of course, are lipid and they don't dissolve in water. There is a glycerol group attached. But anyways, they're hydrophobic, so they don't dissolve in water. So this is the structure of the phospholipid. Here's the chemical structure here. You can see you have your phosphate group right here. These are your 2 fatty acid chains with the attached glycerol. And this one has choline attached, which is an amino acid. So you have your choline, your phosphate, and then you've got your 2 fatty acid tails. So this whole section up here we call the phosphate head, or the hydrophilic head. And the cell membrane itself is made up of thousands and thousands of these molecules. And remember, they're not attached to each other. They're just kind of pressed together and next to each other. Now, naturally, because of the chemical nature of the phospholipids, they will, in water, arrange themselves already into a double layer or into a sphere. Because the lipid repels water, it stays away from the water, and the watery part dissolves into the water, and so it will naturally form a double layer or into a sphere. And because of that, it'll reseal itself, and that's how the cell membrane works. If anything pushes those phospholipids apart, they will naturally come back together, so it can kind of reseal itself. Okay, so here's what the structure will look like. We have our phosphate heads here, and our fatty acid tails here, and here's our double layer. This would be outside the cell, this would be inside the cell. We call this a phospholipid bilayer. And as I said a minute ago, in water, they naturally will form a sphere like this, or a double layer anyway. So you can see already, naturally, these phospholipids will form this kind of a structure in water. Naturally, already, it kind of looks like a cell. Now, remember, the membrane is not just made of phospholipid. It's actually a mixture. We call it a mosaic of phospholipid with some glycolipid. Glycolipid has sugar or a short chain polysaccharide attached. It has sterol in there, cholesterol, and some large embedded proteins. So we call it the fluid mosaic because it can move around, the parts can move around, and it's got a lot of different parts. Membranes are very fluid, as I said. The phospholipids and proteins can move around and drift around in the membrane. Phospholipids also can spin in place, and this all has some impact on how the membrane works, how it functions. So here's a simple diagram of what the cell membrane might look like. We've got the phospholipid bilayer here, and then here depicted in orange are some large embedded proteins, some of the proteins spanning the membrane and some of them not. This particular diagram doesn't show the other parts, so let's look at another one. In this diagram you can see a lot more detail. We've got our phospholipid bilayer again, and it shows some proteins that span the membrane, some proteins that just stick out the top with their attached glycoproteins, or here's an oligosaccharide, so here's our glycoprotein, glycolipid maybe. Here's the glycolipid over here. Okay, so it's a complex structure, but remember, none of this stuff is physically attached to each other. It's all floating around and can move around in place. So let's talk about the different types of proteins that we find in cell membranes. The first type are actually enzymes, and they help control metabolic activities. So in this case, the enzyme would be embedded in the protein, and the active site would be on the intracellular side. So this might be just an enzyme here with an active site that's going to affect some chemical reaction that happens within the cell, and it just happens to be attached there to the membrane. The second type of protein that we find embedded in the cell membrane is a transport channel. Transport proteins span the membrane, and they can transport substances, small molecules, or ions that need some kind of pathway to come through. So if we're looking at water-soluble substances or charged substances, they cannot pass so easily through the lipid layer, so they have these proteins embedded that will allow them to pass through more easily. The third type of protein that's in the membrane are receptor proteins, and we're going to talk more about them next week. But receptor proteins bind with substances outside the cell and cause changes inside the cell. So they'll have some kind of a site here outside the cell where a molecule can interact, and when that other molecule is bound here, it changes the protein shape so that now it opens up an active site on the intracellular side, and it causes some chemical reaction to happen here. Very cool stuff. The fourth kind of protein that's in the cell membrane are cell surface markers. These are recognition proteins. They're kind of like name tags, and they help cells to identify that they belong or that they don't belong, or that they're a certain type of cell. Cells actually have these markers on them, identification markers, and the oligosaccharides that are attached actually serve as those markers. So this is one way that our body can recognize our cells as belonging to us. And finally, the last type of protein that we find embedded in cell membranes are adhesion proteins. And these help cells to stick together. We'll talk more about these next week as well. But one protein in one particular cell and another protein in another cell actually stick together, and this allows our cells to stick to each other. In animals, this is extremely important. We don't want our cells coming apart. So multicellular organisms need these adhesion proteins. Now I'm going to change gears a little bit, and let's talk a little bit about functioning of the membrane. Remember, the membrane controls what goes in and out, and membranes are what we call selectively permeable. I say they choose what can enter and leave. They can't really choose it, but because of their chemical property, only some things can enter and other things cannot, and it's of benefit to the cell the way that things are controlled. Now, very small molecules can easily pass through the membrane. Even water being polar, it can still pass through, because as the membrane is kind of, the phospholipids are moving around, it can squeeze in between because it's small enough. Carbon dioxide and oxygen freely pass through. Glucose, you know that glucose is larger, but it's still not that large compared with some of the larger molecules. Even glucose has to be moved by a transport protein. And of course ions, we already talked about that. So this is a couple of diagrams that illustrate that. Oxygen, carbon dioxide, and water can pass right through the membrane, and do. And it's also represented here. Small hydrophobic molecules. Small uncharged polar molecules, such as ethanol, glycerol, and water, can freely pass through. Larger uncharged polar molecules, such as amino acids, glucose, and nucleotides, are too large to pass through. They're going to need to come through another way. And then anything that's charged like an ion cannot pass through. It's going to need some kind of a channel or a protein. Now, what's going to determine whether or not or how things can move across a membrane is something called a concentration gradient. Concentration gradient represents the difference in the number of molecules of a given substance between adjoining regions. So, for instance, on one side of the membrane versus the other side of the membrane, it's going to be the difference between how many of a particular molecule are on one side versus the other. Substances will move naturally by diffusion from where there's a high concentration to where there's a low concentration, until they reach something called equilibrium. But really it's a dynamic equilibrium, in that the molecules are still moving. Just once they reach that point, the same number of molecules are moving in, as moving out. So it's still dynamic, it's still things moving, but it's at equilibrium, so the inside concentration now represents the same as the outside concentration. So it works something like this. In this case, the concentration gradient comes down this way. We've got many more molecules on one side than the other. I guess this is extracellular. So if they're small molecules that can easily diffuse across the membrane, then they will. So now these molecules are beginning to move across until they reach a point where they're about the same number on either side. The molecules are now still moving, but the same number are moving in as moving out. Now understand, molecules are only affected by their own concentration gradient, so it's not the total number of molecules, but instead it's the number of sugar is affected by the sugar, the number of salt is affected by the salt, etc. Now the factors that affect the rate of diffusion include the steepness of the concentration gradient, so it's going to move faster if there's a bigger difference across that membrane. The size of the molecules, small molecules are going to move faster. The temperature, and you can imagine warmer temperatures, things are going to diffuse faster as well. And then finally, pressure gradients. There's something called the pressure gradient. The higher the pressure, the more things are going to get pushed when you think about it. So high pressure is going to cause a faster diffusion. That's it for this one. We've got two more coming.